Good afternoon, learners. Welcome you back to my session. Now, if you remember, in one of my earlier sessions, I introduced you to the newly launched MSc program of IGNOU in Zoology. And in my earlier session, I gave you a broad overview of the entire Laboratory Course 1 course of MSc Zoology program. So it was just an overview. And then today we will be taking up each of the experiments. So there are as many as 24 experiments in this course, and we will be discussing the different experiments in several sessions, in several Gyan Darshan sessions. So today in the, you can say, the first technical session of Gyan Darshan in the laboratory course, I will take up three topics. That is isolation of plasmid DNA, agarose gel electrophoresis, and quantification of nucleic acids. Why I start with these three experiments? Because these are maybe this is totally new for you, because these are the experiments which are based on molecular biology. Because if in your bachelor's degree you might have done majority of the experiments in classical zoology, like microscopic observation, preparation of cytological specimens, or let's say maybe if you have done some dissections, okay, but now, or visualization of blood samples. But I think that these experiments which I am going to discuss over the coming 2 or 3 sessions, maybe that these experiments could be new to majority of you, or you might not have done it individually, okay, in your undergraduate program. So that is the main purpose. That's why I start with these 3 experiments today. Now, so there are 3 experiments: isolation of plasmid DNA, agarose gel electrophoresis, and quantification of nucleic acid. So we begin with the first experiment, that is isolation of plasmid DNA, and probably these experiments, they are experiment 10, 11 and 12, which you will find in your laboratory manual that has already been uploaded in eGyanKosh. You can find this entire laboratory course manual in your eGyanKosh. So you can download it and you can start studying the manual. Now, first of all, that when we are discussing isolation of plasmid DNA, first of all you should know what is a plasmid. So I will just give you the basic introduction or the principle, and then I will go into the technical part, that is how will you do the experiment. So plasmid, what is a plasmid? Plasmid is a small circular double-stranded self-replicating DNA which is extra chromosomal, which is not present within the chromosome, which is present outside the chromosome, and it is capable of self-replication because it has its own origin of replication, which you will be studying in your theoretical course also. That is, you have one that is paper number 2 which you have, that is the genetics and animal biotechnology. In that course you will study about these plasmids and all, okay, but I am going into the practical part directly. But I presume that all of you know what is meant by a plasmid, which is very elementary. I mean, it's even taught in the undergraduate program. So when introduced into a host chromosome or a host organism via transformation, it will show replication. So why do we need to learn plasmid isolation? Because plasmid is a cloning vehicle. You can clone any foreign gene in the plasmid and you can introduce it into the bacteria. Okay. So for gene cloning, you need to know how to have the plasmid, procure the plasmid, or how to isolate the plasmid. And in majority of the cases, the plasmid isolation is done from a bacterial cell. So there is a bacterial cell where there is already a plasmid, and from there you are isolating the plasmid. So what you need is a bacterial culture, which will have your desirable plasmid. So I hope you get the point what is meant by a plasmid. Now, a plasmid, when you are preparing a plasmid, when you are isolating a plasmid, you have to make sure that it should be free from certain things. If you need to have a purified plasmid, you have to be sure that it is free from genomic DNA or the chromosomal DNA. It should be free from proteins, ribosomes, and the bacterial cell wall. Now, there are several methods for plasmid isolation, of which the most important method which is commonly followed in the laboratory is the alkaline lysis method, where we also use a detergent called the sodium dodecyl sulfate. So that is the alkaline lysis method of plasmid isolation. What is the principle? So alkaline lysis in combination with detergent, that is sodium dodecyl sulfate, is used to isolate the plasmid DNA. So that is the name of the technique which you use for plasmid isolation. Now, what is the principle of plasmid isolation? Using this method, what you do is, the main principle is that you have to disrupt the bacterial cell. You have to break the bacterial cell, because the bacteria, they have cell wall. And beneath the cell wall, you all know that there is a plasma membrane. So you have to break the cell wall. And for breaking the cell wall, you need some very harsh treatment. A strong kind of chemical you need. What is that chemical? That is alkali, high alkali condition, and one detergent, that is sodium dodecyl sulfate. So using these two, high pH and a strong anionic detergent, what you do is you disrupt the cell wall. And then within the cell, you have two different kinds of DNA, because the bacteria will have its own chromosomal DNA and it also has the plasmid DNA, of which what you need the plasmid DNA and not the chromosomal DNA. So what you have to do? You have to ensure that the DNA that you are isolating is plasmid DNA and not chromosomal DNA. And that DNA should be free from proteins, because you know that the DNA, it also has the associated proteins. So it should be free from contamination, because it should be free from proteins or any other matter. And even when you are rupturing a bacterial cell, then there are cell debris, and those cell debris should also be removed if you want to get a purified plasmid DNA. Okay, so since the closed circular plasmid DNA is topologically intertwined, their strands do not separate even in presence of alkaline solution. Lysis steps lead to enmeshing of degraded cell wall, bacterial proteins, and denatured DNA in large complexes coated with SDS. So you are using SDS together with alkaline solution. Okay, and that leads to what? That leads to enmeshing of degraded cell wall, bacterial proteins, and denatured DNA in large complexes coated with SDS. Next, what you do is replacement of the sodium ions in the potassium ions with the potassium ions, because when you are using alkaline lysis, you are using sodium hydroxide. But in the next step, as you will see in the protocol, in the next step, what you do is you replace the sodium ions with potassium ions. We use potassium acetate, and that causes efficient precipitation of these complexes from the solutions. And then you apply a centrifugation, that is in the centrifuge machine, you have to undergo a centrifugation, and then you can recover the native plasmid DNA from the supernatant. So I hope you got the point why we are using alkaline lysis, why we are using alkali solution, why we are using sodium dodecyl sulfate, and immediately after that step, why we are replacing the sodium ion with potassium ion by providing a buffer, let's say potassium acetate. Now, next is, so what are the materials that you require for this experiment? So First of all, you need a bacterial suspension. You need a bacterial culture, overnight bacterial culture. That actually contains the plasmid. So you need to grow the bacteria overnight at 37 degrees centigrade. If it is an E. coli cell, then you can Escherichia coli cell containing the plasmid, which is very commonly used in the laboratory. Then you can grow the E. coli cells in the laboratory condition, incubator shaker at 37 degrees centigrade. Next morning, you come and you remove it from the incubator shaker, and now you have to start your work. Mind that all the equipment should be sterilized. There is an instrument called autoclave where you need to sterilize all the buffers, all the reagents, all the glasswares, plastic wares that you are using. Now, if you take a look at what I am focusing now, that this is known as an Eppendorf tube, where normally we put the culture. This is the Eppendorf tube. So you have to pour the bacterial culture in this Eppendorf tube, because this tube actually can be placed in a centrifuge machine. Okay, and then you can undergo the centrifugation process. Okay, so the centrifuge machine can actually incorporate these kind of tubes, as you can see, and these are called Eppendorf, Eppendorf, Eppendorf tubes. This is of 1.5 ml capacity. Similarly, there are tubes which are of 2 ml capacity. Okay, so mind that you have to take the culture from the culture tube. You have to pour the bacterial culture into the Eppendorf tubes and then centrifuge it. After centrifugation, what you will notice is that you will get a bacterial pellet. Where will be the pellet? The pellet will settle at the bottom of the tube, like here somewhere. If you place the tube like this way, then it will be in the back side. Here you will observe a pellet. What you have to do? You have to clearly pour off the liquid part, because the bacterial suspension is now pelleted, P-E-L-L-E-T, pellet. That is precipitate. It's a kind of, you can say, in a other way you can say it's a precipitate, but we don't say precipitate, we say pellet. You decant the supernatant, because the supernatant is of no use. Okay, so you have the bacterial pellet. Now what you have to do is you need 3 solutions. That is, as you can see, solution 1, solution 2 and solution 3. So the solution 1, it has glucose tris EDTA. This is also, in short, it is called GTE buffer, glucose tris chloride and ethylene diamine tetraacetic acid. So 50 millimolar, 25 millimolar and 10 millimolar. So in that proportion, you have to prepare solution 1. Solution 2 is perhaps the main buffer, because it is the lysis buffer. It contains sodium hydroxide, that is 0.2 normal, and sodium dodecyl sulfate, SDS, 1%. And this solution 2 should be freshly prepared. Solution 1, you can prepare it before an autoclave, but solution 2 should be freshly prepared. That is, when you are doing the experiment, you have to prepare it from the stock solutions, and all the stock solutions should be autoclaved prior to the experiment. Solution 3, potassium acetate 5 molar, pH 4.8, glacial acetic acid and sterile water. So in that proportion or in that composition, you prepare solution 3. So you will need this solution. Solution 3, you place it in refrigerator. Solution 2, never place it in refrigerator, because it contains SDS, detergent. It will precipitate. So don't keep it in refrigerator. Solution 1, you can put it in the refrigerator for future use also. But solution 2 is always, as I said, freshly prepared. Now, procedure. How will you do the experiment stepwise? As I said that you take the bacterial culture in the Eppendorf tube, which I have shown, centrifuge it, okay, you get the bacterial pellet, and then, so the first step is inoculate a single colony of E. coli in 2 ml of Luria-Bertani broth. You need this liquid medium, that is Luria-Bertani liquid medium. Grow the culture till saturation at 37 degrees centigrade and till the optical density is reached. How will you measure this optical density? You will measure it in a spectrophotometer. I will come to spectrophotometer in my third experiment. Then you take 1.5 ml of the culture in the Eppendorf tube and you centrifuge it at 10,000 revolutions per minute for 5 minutes. Discard the supernatant and then you resuspend the pellet in 200 microliter of solution 1. You have already prepared solution 1 and you have to resuspend the pellet in 200 microliter solution 1. Now in this context I will show you how to use this particular instrument, that is which we call as the pipette man, or we call it a micro pipette man. And this is the micro pipette man. Okay, you can set how much. See, there is a particular groove here where there is a it shows how much microliter you are pipetting. So you can move this one and you can rotate this one, you can move this one and you can set this particular. So it will show the readings, how much you are actually pipetting. Okay, so you can change. There are different types of pipette man. This is one of the pipette mans which you are using. And these are called the micro tips. And this micro tip is actually, you can attach it like this. So, and here you have to press it gently so that you can actually pipette out some liquid. Like I am showing you how to use it. Like this is the Eppendorf tube. Let's say there is a particular solution here and you open the tube like this. See, this is open now. This is the lid. And then you just dip your pipette man inside and then slowly you just press it up like this. See, it contains the solution. So I have pipetted out the solution from the Eppendorf tube. So if you need to add any solution, 200 microliter, how will you do it? You have to set the 200 microliter here and then you have to add, like let's say there is the bacterial pellet here. So what you have to do, you have to gradually add the solution like this. See, it is being added. So this is the way by which, because this instrument you have to use very regularly when you are doing such experiments, and you have to have a control over this instrument or over this pipette man. So that is the way I am showing you again. So you can just pipette it out like this. Okay, so you have the solution. Make sure that there are no air bubbles. You should pipette it out very gently. And then when you put some solution inside, again, you have to do it very gently and you have to resuspend the pellet in this solution. Let's say this is solution 1. So I hope you got the point that how you have to use this instrument. You have to be careful and handle it gently, because these instruments are costly. Some of them are quite costly, and if it is damaged, then again it needs repair, and for that you have to it needs some expenses. So you have to be careful while handling these instruments. So you have resuspended the pellet in 200 microliter solution 1 and then leave it for some time on ice for 5 minutes. In the next step, what you do is you add 400 microliter of solution 2, freshly prepared solution 2. So initially you have added solution 1, that is GTE buffer. Now you are adding solution 2. You mix well several times. How will you mix? Just look at how I'm mixing, like very gently, like this. You will mix it. So the pellet was dissolved in solution 1, then in solution 2, and then you are mixing the solution like this. Okay? And what is the utility of this step? The utility of this step is it contains SDS and alkali, sodium hydroxide. So SDS solubilizes the phospholipid and protein components. Sodium hydroxide denatures the DNA as well as the proteins. So you don't want the chromosomal DNA, you don't want the proteins. And leave this again on ice, or you can leave it on room temperature also, not a problem, for 5 minutes or 10 minutes. You can leave this for 10 minutes also. Third step, that is, now you have to add solution 3. This is also ice cold. 300 microliter of solution 3. Again you mix well as I have shown you. Incubate on ice for 5 minutes and then you have to centrifuge it. Now what is the utility of this step? This step contains potassium acetate. And this potassium acetate, it neutralizes, because there you have used high alkaline solution. So you need to neutralize it now. Once the disruption of the cell wall is over, you have to neutralize the solution. And you are adding potassium acetate because here the pH is 4.8. So it is an acidic solution. So you are neutralizing the sodium hydroxide in the previous step and you are precipitating the genomic DNA. The glacial acetic acid lowers the pH to 4.8, making high salt in low pH with the chromosomal DNA entrapped in the coagulated protein. Again you mix it as I have shown you and then you centrifuge it. Centrifuge it to high speed, 12,000 revolutions per minute in the centrifuge machine for 5 minutes at room temperature. Now make sure that you have to collect the supernatant, not the pellet. You have to discard the pellet or the precipitate. You have to collect the supernatant. And you collect the supernatant to a fresh Eppendorf tube. So you have to again use the pipette man to collect the supernatant. And it should be clear supernatant. There should be no cell debris. There should not be any impurities. Otherwise, your plasmid preparation will be impure. So it should be clear. It should be free from cell debris. And you collect the clear lysate in a separate Eppendorf tube. So up to this is done. Now the third step is the purification of the plasmid DNA. That is, how will you further purify? Already it is to some extent purified because you have removed the proteins. You have removed the chromosomal DNA, but it needs further purification. And for that, what you do is, this solution is subjected to treatment with phenol chloroform. That is equal mixture, 1:1. Like if you are preparing 30 ml phenol chloroform mixture, it means 15 ml of phenol and 15 ml of chloroform. But mind that this phenol should be equilibrated. The pH of the phenol should be of pH 8. How will you do it? You have to first equilibrate phenol in such a fashion that the pH is 8. And many of the companies, they actually sell trace equilibrated phenol. So that is not an issue. You can just, any study center where there is the laboratory and they are undergoing training for the students, undergoing training program, practical programs for the students, they must have this, or they must purchase this trace equilibrated phenol. And using that phenol and chloroform, you prepare this phenol chloroform mixture 1:1 and you add it to the supernatant. That is, if your supernatant is, let's say 1 ml, you have to add equal volume, that is 1 ml of phenol chloroform mixture, and then centrifuge. Centrifuge machine should also be handled with care, and being the course coordinator of this program, I have already mentioned it in your laboratory manual that you should be very careful in handling the instruments. Until and unless you are 100% confident, don't use the machine on your own. You ask your counselor, whoever is there, to show it or properly demonstrate it before you directly handle them. Okay, so this has to be kept in mind. Don't get super excited by seeing the different instruments and start handling them without knowing the operation procedure, because these are very, very costly. It costs in lakhs, some 5 lakhs, 4 lakhs, and if the instrument gets damaged, your total practical experiment will come to a standstill. So be careful. Okay, so centrifuge it at 12,000 rpm for 10 minutes at room temperature. Collect the aqueous layer, that is upper aqueous layer, and then add chloroform again. Again you centrifuge. Now why we are using this phenol chloroform step? Because phenol again is another, it is another way by which you can remove the associated proteins. And why you are using chloroform? Because chloroform, if there is any phenol contamination, then it might damage the DNA. So you need to remove the phenol also. There should be no associated phenol. So you further try to remove that phenol by adding chloroform alone in the next step. Okay, so this is done. Now what you have to do is, the phenol chloroform step and the chloroform step is over. Now you take the supernatant and you precipitate the plasmid DNA. How do you precipitate the plasmid DNA? You add sodium acetate pH 5.2 and chilled absolute ethanol. So if your supernatant is, let's say 1 ml, you have to add 1/10 volume, that means 100 microliter of 3 molar sodium acetate pH 5.2 and 2.5 volume or twice volume. That means how much? 1 ml plus 100 microliter, that is 1 ml 100 microliter, 2 volumes of that. That is, it can be even 2 ml 200 microliter, twice of that volume. That is chilled ethanol. And in this step actually the plasmid DNA will be precipitated. You have to keep the mixture at minus 20 degrees centigrade for 30 minutes or for 1 hour also you can keep. And then again you have to centrifuge. When you centrifuge, what will happen? Now the plasmid DNA, actual plasmid DNA will be pelleted. So now in this step what you have to do is you have to remove the supernatant. You have to discard the supernatant. And you have only the plasmid DNA which is being pelleted. Next step what you do is you wash it with 70% chilled ethanol. Why? Because you want to make this plasmid DNA free from contaminating salts. There should be no contaminating salts associated with this plasmid DNA. So you have removed that protein, okay, you have removed the chromosomal DNA, and now you are making it free from salt, so that you get a very, very purified form of plasmid DNA. So washing means you have to again centrifuge it at 12,000 rpm for 5 minutes. You can repeat this step twice. Discard the supernatant, and now what you have to do is you have to dry the pellet. So now the final step is you have the pellet in the Eppendorf tube and you dry it, and now you dissolve it. You can dissolve it in water, sterile water, or you can dissolve it in TE buffer pH 8, that is tris EDTA buffer pH 8. And therefore now the plasmid DNA pellet is being dissolved in TE buffer or in sterile water. So that completes your total plasmid isolation. Okay. So the main principle I hope you have got, that you have to lyse the bacterial cell. Okay. Then you have to lyse it using alkali SDS, neutralize the solution, undergo centrifugation, collect the supernatant, purify the plasmid DNA through phenolchloroform and chloroform extraction, and then finally precipitate the DNA using 1/10 volume of sodium acetate, pH 5.2, and twice the volume of chilled ethanol. And then you get the pellet, wash the pellet with 70% ethanol, and then you dissolve it in sterile water or TE buffer, pH 8. So you have the, like if this is the Eppendorf tube, as I have shown, you have the DNA solution inside. That is, the DNA pellet is dissolved inside, and that is present in this tube now. So this completes one experiment. Second experiment is agarose gel electrophoresis. That is, you need to visualize. I will come to the observation part, that is how will this plasmid DNA look like after I discuss agarose gel electrophoresis. Because you will need this experiment, agarose gel electrophoresis, to visualize the plasmid DNA. Now, what is agarose gel electrophoresis? It is a method used for the separation of nucleic acid fragments and analysis of DNA restriction fragments based on their size. It is one of the most widely used techniques used in biochemistry and molecular biology. This technique has been used in the detection and characterization of plasmid DNA and chromosomal DNA. So you need this technique to see the plasmid DNA. How the plasmid DNA will look like, that you have to see, you have to visualize. Whether you have isolated properly or not. How will you check that? You will check that through agarose gel electrophoresis. So this is the immediate experiment that will be followed after plasmid DNA isolation. Now what is agarose? Agarose is a linear polymer composed of alternating residues of D and L galactose joined by alpha-1,3 and beta-1,4 glycosidic linkage. There exists an anhydro bridge between the 3 and 6 positions of L-galactose residue. 800 galactose residues per chain are present in commercial agarose polymers. So basically, it is actually nothing but these are residues of alternating residues of D-galactose and L-galactose, and they are joined by the glycosidic linkage. And you are using this agarose as a matrix for visualization of DNA molecule. It can be either plasmid DNA, which we are discussing today, or it can be chromosomal DNA also. Now, what you have to do in this case, or what is the principle of this technique? That is, the DNA migration, when you are actually loading the DNA in an agarose gel, it is regulated by several factors. One is agarose concentration. That is, there is a linear relationship between logarithm of the electrophoretic mobility of DNA and the gel concentration. So if the gel concentration is high, mobility will be lower. If the gel concentration is lower, mobility will be faster. Agarose type, that is, what is that nature, whether you are using a normal agarose or a low melting temperature agarose. It depends on the type of agarose. DNA conformation. Whenever you are visualizing a plasmid DNA in an agarose, there are 3 different forms: superhelical circular, nicked circular, and linear form. So normally the plasmids will come in these 3 forms in an agarose gel, which I will show later. DNAs migrate through the agarose gel at different rate. If the molecular weight is high, they will migrate slower. If the DNA molecule is of smaller size, they will migrate faster. Like you have a DNA of 500 base pair and you have another DNA molecule which is 1 kilo base pair. So 1 kilobase pair DNA molecule will migrate slower because it is of high molecular weight. It is larger in size, and the 500 base pair DNA molecule will migrate faster because it is smaller in size, low molecular weight. So that is also another factor. Electrophoresis buffer, that means when you are running the DNA in an agarose gel, you have to use a running buffer which is present in the electrophoresis tank. And the composition and ionic strength of the buffer, it also affects the mobility rate. If it is a high ionic strength, then efficient conductance will occur, but a large amount of heat is generated. So you have to actually properly balance. You can, if you are using a very high voltage, okay, in the electrophoresis system, then the DNA will migrate faster, but then the resolution may not be good. If you are using a very high current, a lot of heat will be generated, and because of that heat generated, the DNA molecule might suffer some damage. It might suffer some heat denaturation. So you have to properly balance. Don't use an excessively high voltage in the power supply, or don't even use a very low voltage also. That is, you have to use a moderate voltage. Normally, we use 70 to 80 volts for running the DNA. Or if you are measuring in terms of current, then it could be within the 20 to 30 milliampere range. But normally we run the DNA at a constant voltage, which is more or less in the range of 70 to 80 volts. So there will be a power supply system where you will actually use, which you will actually use for running the DNA. So you have to cast an agarose gel. I will show the picture, and then load the DNA and then run the gel in an electrophoresis buffer, and where the electrophoresis tank is being connected to a power supply where a fixed voltage is being set. And because the DNA is negatively charged, the migration will be from the anode, from the negative pole to the positive pole. Because the DNA is negatively charged because it has an exposed phosphate group. So mind that the migration will be from the negative to positive. And so when you are running the gel, you have placed the gel tray within which you have loaded the DNA sample in the electrophoresis tank. It should be on the upper portion of the gel should be towards the negative polarity, and from negative to positive it will migrate. So be careful when you are running the gel and you are connecting the gel to a power supply system. Ethidium bromide in the gel. Now what is the function of ethidium bromide? Ethidium bromide is an intercalating agent. And we normally use ethidium bromide to visualize the DNA under UV transilluminator. So you have to include this, you have to stain the DNA molecule using ethidium bromide. So you can use ethidium bromide in the gel also, or you can separately, stain the gel separately after running the DNA. Or you can add ethidium bromide when you are preparing the gel. That also you can do. So there are options, but if you are incorporating in the gel, then this is one of the factors that the negative charge of the DNA is decreased, and there is a slowing of the migration rate of the DNA dye complex by a factor of 15%. So ethidium bromide is used for visualization of the DNA. What are the electrophoresis buffer that you are using? That is tris EDTA pH 8 or tris borate, that is TBE, that is also pH 8. So you have to use this running buffer. This is actually the running buffer, electrophoresis buffer. That is, there is an electrophoresis tank within which you will pour this buffer and then place the agarose gel or the agarose gel containing tray within this electrophoresis tank, and then run the gel. Load the DNA and run the gel. Now just take a look at the this thing, that is the picture. So you can see that this is the first picture. It shows a glass plate, and that is the gel casting tray. Second, it shows that you have to actually properly seal it so that there are no leakage. Okay, so you are using it, tape, masking tape, to wrap up the two ends of the casting tray. And then what they are showing? They are showing that you are pouring the gel. So what you have to do, you have to prepare the agarose gel. You have to prepare it in the TAE buffer. That is, you measure the agarose. Normally we use 1% agarose. So you have to prepare 1 gram of agarose. You have to weigh 1 gram of agarose. And then you have to dissolve it in TAE. That is 1x TAE. So you have to prepare tris acetate EDTA buffer. Okay. And this is actually the running buffer. So you dissolve this agarose in 1x TAE by properly melting it. Okay, so you have to boil it so that the agarose completely dissolves. And after that, you have to pour the agarose gel and allow it to solidify. But before it gets solidifies, you have to place a comb, as you can see in the third. You have to place a comb because you have to create the wells. And after the gel has solidified, you remove the comb so that in number D you can see the wells are created. And then using that same micropipette, you have to load the DNA sample. But you have to mix the DNA with a tracking dye. Gel loading buffer. You have to use a gel loading buffer, and you have to mix the DNA with the gel loading buffer. I am coming to the composition of the gel loading buffer, and you have to add the DNA. And then in the last step they have shown, as I said, that you need to connect the gel tray with the power supply system. That is, it will migrate from the negative to the positive, that is from negative polarity to the positive polarity, because DNA is negatively charged. So now the migration will occur. DNA will gradually migrate into the gel, within the gel, and it will migrate from the lens and it will migrate within the gel. And you have to continue this running for some time. Maybe 30 minutes to 1 hour at least you have to run the gel, and then you have to stop the power supply, take out the gel tray and stain the gel. Where will you stain the gel? You will stain it in ethidium bromide. Ethidium bromide is an intercalating dye, and after staining with ethidium bromide, if you place this under UV transilluminator, then it will show fluorescence. And that will enable you to visualize the DNA band or the plasmid DNA. So in between, there were some steps. Now if you come back here, that we discussed on what are the factors affecting the migration of DNA in an agarose gel. We have discussed what kind of electrophoresis buffers you can use, and the tracking dye that I was talking about, which is present in the gel loading buffer, contains actually bromophenol blue and xylene cyanol. Bromophenol blue is a dye, xylene cyanol is also a dye. Now these are tracking dye. That means how long will you run the gel? That will be dictated by the presence of these dyes, because migration rate of bromophenol blue is 2.2 fold faster than xylene cyanol and corresponds to 300 base pair DNA, while that of xylene cyanol corresponds to 4 kilobase pair DNA. That means where you will find the bromophenol blue will signify that is more or less 300 base pair DNA. And the region where you will find the xylene cyanol dye within the gel, you will understand that is more or less 4 kilobase pair DNA. Now you know what is the size of your plasmid, and accordingly you will run the gel. I mean, you will keep notice on these two dyes in the gel, and accordingly you can plan how much time will you run the gel. Make sure that the DNA does not come out of the gel in the electrophoresis buffer. Then you will not be able to visualize the DNA. Okay. So that is the importance of this tracking dyes. And with regard to the fluorescence, I already told you that the ethidium bromide that you are using. So you need, what are the materials that you need? Agarose. As I told you, you need to dissolve it. Tri-CDTA, which is the running buffer. So there is the composition. I am not going into composition. You will find it in your manual. Then you can use tris borate EDTA also, TBE buffer. And gel loading dye, that is bromophenol blue, xylene cyanol, glycerol. In place of glycerol, you can also use sucrose. DNA staining dye, ethidium bromide solution, which is 5 mg per ml. And you need a DNA ladder also. Why a ladder? Because the ladder is nothing but it's a molecular weight marker. Because if you want to know the size of your DNA, supposing you are not sure about the size of the DNA, then there is something available called the marker. And this marker actually consists of different sized fragments, different sized DNA bands. Like lambda DNA, digested with Hindi3. It gives some specific fragments of specific sizes. And that you can load in the agarose gel, side by side. And you match your DNA along with respect to the molecular weight marker, to which band it corresponds to. And then that will give you a rough idea regarding the size of your plasmid DNA or the size of your DNA, which you have isolated. So just comparison. Comparison with a ladder means comparison with a series of fragments whose molecular weight are known and which you have loaded in the agarose gel. And then of course you need the electrophoresis system, the electrophoresis apparatus, the power supply. These are also needed. Gel sealing tape. And mind that, again I repeat, all the chemicals, glasswares, okay, and the buffers, these need to be sterilized. Of course, you cannot sterilize the electrophoresis system. I am not talking of that. Not the gel casting tray or the electrophoresis system. That you cannot autoclave. That you have to wipe it clear with alcohol, ethanol. So that will sterilize automatically. But for the other reagents that you are using, you have to prepare them in a sterile container using sterile water, because these are all molecular biology work, microbiology, molecular biology work, cell culture. These have to be done under absolute sterile condition. There should be no contamination, because otherwise your plasmid DNA will degrade. If there is DNase contamination, then DNase, what it will do? It will degrade your DNA. You will not be able to find a very good quality DNA. You will get a smear. So that is not expected after such a laborious procedure. So you should be careful. You should maintain cleanliness. You should maintain the sterile condition. Now I am coming to, as a result of these two experiments, finally what you will be able to see is a plasmid band. And if you see the observation, go back to the first experiment, you can see here that on the right hand side of the agarose gel, this is the photograph taken. Okay, you are visualizing the DNA under UV transilluminator, and you have taken, one has taken the photograph. So you see that mostly here you get three bands. As I have already said, one is the supercoiled plasmid DNA band, which is the fastest. Above that you will get the linear plasmid band, and above that you will get the open circular plasmid. And if there is some genomic DNA contamination, you might observe some genomic DNA contamination, although it is not expected. Okay, but sometimes even after such procedures, you might find some genomic DNA contamination. Okay, but you are expected to find mostly the three different forms of plasmid DNA, that is supercoiled, linear and open circular plasmid, of which supercoiled band is the most prominent. Even if you are not able to observe the other bands, the supercoiled plasmid band will always be visible. It is the fastest migrating and it is also of sufficiently high concentration. It comes usually in high concentration. So there are several precautions. Those are also given in your manual. I am not reading it out. Please go through the manuals that what kind of precautionary measures you need to take for these two experiments. The last experiment, that is the quantification of nucleic acid. What is meant by quantification? Quantification means I need to find out how much DNA has been isolated. I mean, per ml, how much microgram or how much milligram is present. How will you do that quantification? And how will you estimate the purity of the DNA? That is done through spectrophotometric measurements. You have to measure the sample, you have to measure the absorbance of the sample in a machine called spectrophotometer at 260 nanometer and 280 nanometer. And you have to find out the ratio, that is A260 by A280. This ratio will give you an idea regarding the purity of your DNA. Now I am coming to that what should be an ideal ratio. So what you have to do here, and this entire what should I say, philosophy or entire principle is actually based on a principle called Beer-Lambert's law, which is a very well-known principle in biophysics, especially when you are undergoing the UV visible spectroscopy. And the guiding principle is the Beer-Lambert's law, which shows that I equal to I0 10 to the power minus epsilon dc, where I is the intensity of transmitted light, I0 is the intensity of incident light, epsilon is the molar extinction coefficient, d is the optical path length, that is you take the DNA sample in cuvettes, quartz cuvettes, and this cuvette has a path length which is taken in centimeter, and C is equal to the concentration of the absorbing material. Okay, so using this principle you have to calculate. Actually you just take the sample in the cuvette and you have to place the cuvette in the spectrophotometer, and the spectrophotometer will give you a reading at 260 nanometer wavelength and 280 nanometer wavelength. So you have to take two readings. So what you need here is a spectrophotometer, quartz cuvette, DNA or RNA sample. In this case, in our case, it will be DNA sample only, because you are not working with RNA. You are dealing with the plasmid DNA. TE buffer, pH 8 and the micropipettes as I have shown. So what will be the procedure? Now first of all you have to standardize, or you have to you have to auto zero the spectrophotometer. So for that what you have to do is you have to take a quartz cuvette within which you will take simply the buffer, that is 3 CDTA buffer, and calibrate the spectrophotometer. Like in a pH meter also you have to calibrate a pH meter. So you have to calibrate the machine first at 260, that is you are just doing it auto zero. So at 260 nanometer and 280 nanometer. And then you add the samples. I mean you discard the, you have two actually sectors or two blocks. I mean you have to place two cuvettes. So you use another blank which is also the buffer. Don't throw it. You have to keep it as it is, that is the blank, and against that blank you are measuring the absorbance of your sample. So you take the DNA sample which you have dissolved in Tris EDTA buffer and you measure the absorbance of the buffer. So this buffer will act as a blank against which you are measuring your absorbance. So now you note the OD values, that is 260 nanometer and 280 nanometer, and you calculate the OD 260 OD 280 ratio. Now ideally, if your DNA is pure, then the OD 260 OD 280 value is supposed to come to 1.8. I'm not talking of RNA because right at the moment you might not need, but just you can have it for your knowledge that for RNA it will be 2. And for DNA, which you are doing the experiment, for that it will be 1.8. Any deviation from this value will give you an idea that there is some sort of error or your preparation is not that good. Okay. So from this value 260 to 280, it is very important that you get an idea. And OD equal to 1 at 260 nanometer means 50 microgram per ml. So from that you can also calculate what is your OD value at 260 nanometer. If it is 0.5, then it should be half, that is 25 microgram per ml. So accordingly, what absorbance you have got, based on that you can calculate what is the concentration of the double-stranded plasmid DNA that you have got. So two things are there. One is A260, A280 ratio will give an idea regarding the purity of the DNA, and A260 nanometer value that you have got will give an idea regarding the concentration of the DNA. So using the same experiment, you can get two information. So that is actually the principle of this. So you can accordingly note down your observation in this table form, that is nucleic acid sample OD260, OD280, and then measure the ratio, and from the OD260 value you can calculate the concentration of your DNA. So I think this was the point of discussions that I had today. That is the 3 experiments of your laboratory course, that is experiment 10, 11, 12. Please go through the manuals. Everything is very detailed out there, and the protocols are very widely, elaborately discussed. Figures are also given, and you need to have a very good idea regarding the principle of the technique, because in the Viva Voce also later on, after the completion, you may be asked several questions. So please go through the principles, and in case you find any difficulty, you are always welcome to send your queries or to contact us with your questions. But this is a newly launched program of MSc in Zoology, so you have to be very, very careful because you are the first batch. Okay, your performance will actually dictate the success of this program. So we carry a lot of hope in terms of your performance. Okay, so that is it for today. In my coming session, next laboratory session discussion in May, I will be taking up another 3 experiments. Okay, that also on molecular biology, and we will be discussing on those 3 experiments. Till then, please go through these experiments, and please have a very happy learning. Thank you.